This demonstration shows the technique for removing the germline precursor cell from early embryos in the Amphipod Crustacean Parhyale hawaiensis. Parhyale is an emerging model organism with expansive potential for use in evolutionary and developmental research. Most model arthropods are insects, and the most extensively studied of these is the fruit fly Drosophila melanogaster. Insects are nested within the subphylum Pancrustacea, and studying other crustaceans outside of the insects provides a broader view of the evolutionary history of the developmental traits and molecular mechanisms that have been well studied in Drosophila. However, very few crustaceans have been well established for experimental laboratory analysis of development. Parhyale is a highly tractable laboratory model system, and thus represents a valuable addition to the inventory of model organisms. As a laboratory animal, Parhyale is very easy to maintain and work with. It can survive well in large cultures of artificial seawater, and it is easy to distinguish between males and females based on clear morphological differences. Most notably, the large hooked anterior appendages that males use to grasp the females during mating. For developmental work, Parhyale has several very appealing features. Couples mate all year round, so it is possible to gather embryos at any time. Females lay embryos into the brood pouch between the first several pairs of legs, and it is possible to gather the embryos very early in development without killing the female or damaging the embryos. The embryos survive in filtered artificial seawater through hatching, and there is a detailed staging table that